Hi, in this video I am going to go through the differential expression analysis and final project poll from week 13 of spring 2024. With that, let's get started. The first question has you go through different RNA types just as a refresher of what they are. So the first one says contains introns. tRNA, microRNA, and ribosomal RNA are all non-protein coding sequences. And unless maybe they occasionally end up getting translated due to some reason, they shouldn't have introns. Mature mRNA isn't at least notably going to contain introns, although sometimes from random processes an intron might slip through, or a section of RNA may only sometimes be an intron and may sometimes be an exon due to, again, it's all chemistry, so it's a little bit random at times. However, immature mRNA, so immature messenger RNA, is definitely notably going to contain introns. When it's immediately transcribed from the DNA to the RNA, it will contain those introns initially. Transfer RNA is, by definition, going to be the RNA that brings amino acids to the ribosomes. That's it, that's just their definition. Mature mRNA has a polyadenylated tail added to it that allows it to exit the nucleus, and then this polyadenylation is actually one of the primary molecular tools that we have to isolate specifically mature messenger RNA. And so it actually is used as part of the isolation process for collecting messenger RNA from sequencing data. You can just separate it from the immature. Non-coding sequences often involved in RNA silencing and gene regulation. That is our definition of microRNA. They also tend to be short sequences, but they're non-coding. That's kind of the special thing. Finally, comprises 85% of all RNA in human cells by mass. That is ribosomal RNA, which is that weird protein and nucleotide complex that actually does the translation and builds the amino acid chains out of, or based on, the messenger RNA template. And they are hugely abundant. It is the vast majority of RNA. If you wanted to isolate specifically messenger RNA, and that's like from a bunch of lysed cells, that's going to be proteins and carbohydrates and fats, as well as other nucleotides such as immature messenger RNA. It's going to have ribosomal RNA. Definitely got to get rid of that because that is a huge amount of the RNA. And of course DNA as well. So a way to isolate mature versus immature is to isolate sequences of polyadenylated tail. That by itself is a really major improvement in this technique. And if you only isolated sequences perfectly with these poly A tails, that's probably going to mean that most DNAs, I think all ribosomal RNA, is going to be removed as well. But ideally, you're also removing them. In terms of adding RNase, that is going to break down your RNA. You don't want to do that. Adding DNase is basically going to be removing DNA, and so that will remove those. Another thing that's not really in this list is then removing those degraded nucleotides, although a lot of the different processes that you do when isolating RNA end up also removing those degraded nucleotides. Adding chemicals that denature proteins is a great move. It helps prevent the active degradation of RNA. RNA has a limited half-life in our cells, sometimes on the order of minutes, but often around minutes to hours in human cells, although in other organisms that operate on a much faster time scale in terms of a single cell, it can be on the order of seconds to minutes. It can be quite big, quite fast. Adding chemicals to protect proteins. I don't really want to protect the proteins. I want the messenger RNA, so I don't really need to protect proteins for any reason. Do not freeze as long as possible. No, it's the opposite. Keep frozen until the last moment possible. You really want those mRNAs to represent like an instant in time typically. And so you're really wanting to keep them frozen. Oftentimes you're collecting directly into liquid nitrogen. I asked you to guess what the average half-life of mRNA molecules in living cells are. It's approximately minutes. Guess the lifespan correlates the lifespan of the protein. It's a strong positive correlation. That it's some positive correlation, I believe. So the lifespan of the protein is going to tend to be much longer than the lifespan of the mRNA. The quantity of the mRNA should also correlate with the quantity of the protein, but these are not the same thing. So this is where we see a strong positive correlation. In fact, I think this one may be little correlation. The protein may last indefinitely, and the mRNA is always going to be fairly limited. Okay, and when we get down here, we see that the mRNA half-life, we see extremely little correlation in R squared of 2. So that is a coefficient of the percent of the variance explained by a linear model. It's not the coefficient, it is the percent of variance explained by a linear model. So only 2%. So definitely a very, very poor predictor of protein half-life. Now, on the other hand, the R square here is 41%, which is saying that our mRNA copy strongly correlates with our protein copy. I've also shown here the diagrams just to show that, you know, we are actually kind of meeting tests for correlation parameters. These are both normal distributions, although they definitely have different variances overall. So it's not perfect, but it's pretty good. So mRNA and protein half-life do not obviously correlate. Copy number and protein copy number clearly correlate. And for the record, if you're ever unsure, can you draw a sloped line through the distribution? That's really what you're looking for. Here, any distribution, any line that you're going to draw is going to have a very, very slight slope. And even against that slope, there's going to be a huge amount of error. Whereas here, that's not true. It does seem to be a little bit less of a linear relationship, which is kind of interesting. I'm sure we can get into some chemistry discussion on why that would be. Pretend you are building a new reference genome for an uncharacterized species. If you want to sequence a species, I don't think I did that correctly. I think there should be another S in there. Species' mRNA and see all or like 99% of the genes within that species genome. What kind of sequencing strategy do you think would be appropriate? You would definitely want to see many different individual genotypes, but the much more important selection is the many different types of tissues. So really, if I want to rank these, select as many different types of tissues as possible. That's going to be one of our main differences between tissues, is what genes are being expressed. There are plenty of genes that are expressed in all living tissues because they're necessary to maintain homeostasis. But there's also a large number of genes which are specific to only a small number of tissues, or maybe a single type of tissue, though that's going to be less common. Differences between tissues are going to be much more often like the combination of different genes as opposed to any single one. But certainly some genes are very specific. Sample many different time points / developmental stages. That's probably your second most important of these. And then of like these sampling issues, the sampling different genotypes, because the vast majority of individuals are going to have the vast majority of the genes the same. But many times genes will get turned on or off at different time points. Using PCR reactions to amplify specific sequences. There are ways to allow that to make it work, but that's not really what we're going for. So like there are ways to take advantage of PCR reactions in RNA sequencing, but oftentimes you really want just as perfect of a snapshot in that time as possible. And if you bring PCR into the mix, you start to cause further issues because not all RNA sequences are going to amplify as reliably as others. So that can actually be kind of dangerous and detrimental to an analysis. Finally, isolating messenger RNA is the whole point, and so that definitely should be true. I don't need to isolate proteins. I guess isolate them away, maybe, if that's how you interpret it, but that's not what I was getting at. In the below plot, what comparison could I make to generate a logical object capable of removing the point to the right of the blue line? Pretend the data containing the plotted data frames PC1 and PC2 is located inside of an object called PC1 $X. So if this by itself is a data frame, the output of PR comp is a data frame inside of a list. So this would be the data frame inside of the PCA1 list object, which is the output of PR comp. In that scenario, I'm extracting PC1, so I need to do something that allows me to PCA1 $X. I need to first extract that data frame. Then I need to extract the first column. I could also have, assuming it was made by PR comp, I could have done this, where I was extracting it by name. However, the much easier way is simply to do that. If I am then testing that value and trying to see if it is this or this, capable of removing the point. So if I want to remove that point, I need logic that selects only these points and omits this one. So if I say that this x-axis is, say, less than, really anything less than about 105 or 10 or so will work. I'm just going to do 300 because that's what I did in this diagram. This will produce a lot of trues. Most of these are less than 300, but this one is not. That would then allow me to use that object in order to do a subsetting. And there we go. So just breaking this back down. Here is my logic. Did I ask for the logic object? Yeah, so I asked for the logic object. This is the actual answer. The rest of this is how I would then use that logical object to subset everything but the data. Now, the interesting thing, and this is actually a big issue with students, is they would try to subset PC1 by itself. But that was never the point. The point of subsetting PC1 in the pipeline was so that you could modify the pipeline object, not the PC1 or PCA object. I don't care about the PCA object once I've done this visualization, but I definitely care about using it as part of the analysis. Very common source of error. So in summary, that is the logical object you would need, something like that. Had you flipped it, this would have been still valuable, but I would need to do something. So I would, when I'm using it in extraction, I would need to like flip the logic or something like that. So this is the better choice. It's directly usable. In the following plot, I use color to indicate overall expression for the gene. However, something is strange about the color gradient scale. What is uncommon about this scale? I can see that it is going up by powers of 10, which means it's going up, like this. A very, very fast trend. It's a logarithmic trend, and that's what we would call that, is that it is scaled logarithmically. Wow, just pretend I spelled logarithmically. Probably close to right. Specifically, it is using a base 10 log. And I can tell that because of how it's done the rounding here, and this is just a feature of ggplot, that if you scale the gradients logarithmically, you often will get these prettier demarcations for different values. So just a nice little fact. So if you got a bunch of really ugly numbers, you probably used a natural log, which is just log, or maybe even a log 2, which is log 2. But log 10 is log 10, all one word. Trans equals log 10 was used as an argument specifically. All right, in the below plot, how would you describe the expression of Muc16 in my group, tumors without APC mutations relative to samples from tumors with high impact mutations? So I'm trying to focus in on the tumors without APC mutations. Let's go look at that plot. I know it said below, I'm sorry. So here, the definition of the x-axis is this minus this, which means that if Muc16 is negative, this must have had a larger value than this one. So relatively, that produces a negative, and so this must be upregulated relative to the relatively downregulated high-impact mutations. So upregulated in group 1 and downregulated in group 2. And the reason you know that is based on the order. Now here I type in the order, but specifically it is group 2 minus group 1. That's the really important part. Whatever you define your group 2 and group 1 is, that's going to be what determines this, but that's how I define mine. And that's fairly typical. The less like wild type compared to the more wild type. So group 1 being more wild type, group 2 being less. It's all very circular, but that's how you go through it. And that's it. Hopefully that helped in terms of the tests, you know, being able to interpret these, at least understanding how PCA component analysis works and is interpreted. I think there will be very little on this isolation since we never talked about it in class, although I might just give you this picture. I like this picture. I think it's a good learning tool. Anyways, have a good day.